Today, we shall discuss about gene mutations that forms an integral part of genetics and molecular cell biology and helps us understand the underlying mechanism of gene expression. We will discuss in detail about what mutations are, the different types of mutations, what causes these mutations, what are the consequences of such mutations, and what are the mechanisms that are in place in the cells to help repair such mutations. With respect to genetics, life is nothing but the perpetuation of genetic material. That is the passage of genetic material from parent to offspring or from parent cell to daughter cells. The genetic material present in the cells needs to be copied into another set, which is then divided into daughter cells. This takes place with the help of the replication machinery in the cell. The crucial need for such a propagation is the maintenance of the genome through many cycles of replication. During replication, there is possibility of an error or the genetic material being damaged, which can then be passed down to the daughter cells. The daughter cells carry forward such changes to the genetic material, which is called as mutation. So mutation is defined as a permanent change in the genetic material. And the consequences of mutations depends on the site of mutation. That is where the mutation is taking place. At an organismal level, that is at the level of an organism, if the mutation takes place within a somatic cells are the cells that undergo mitosis in the body. It is called as somatic cell mutation. This mutation affects only the individual carrying the mutation. Example of such a mutation is lung cancer caused by smoking. So if a person has lung cancer due to smoking, that means he carries a mutation in the lung cells that has caused the cells to become cancerous. Now this cancer is not transmitted to his children because it is not a germ cell mutation. Coming to the other one, if the mutation takes place within a germ cell is a cell that undergoes meiosis in the body. So it is called as germline mutation. This mutation affects all descendants carrying the mutation. Example of such a mutation is baby born with birth defects to parents who were exposed to radiation or carcinogens. So this is a result of germline mutation. So the germline mutation has been carried to the children who, if they have children, will also carry the mutation. At the cellular level, mutations can occur in different sites of the genome. For example, we'll discuss on how glucose uptake is controlled by the genetic expression of insulin gene. Now let's consider this is part of a genome where insulin expression is being controlled. And this is the gene responsible for insulin production. So any gene will have a promoter region which controls the expression of its gene. So whenever there is a signal from outside the cell to produce insulin, an RNA polymerase comes and binds to the promoter region. After binding, the insulin is transcribed into mRNA. The mRNA is then translated using ribosomes into protein. In this case, it's insulin. So this is how insulin is produced. Once there is a signal from outside the cell for insulin production, the RNA polymerase binds to the promoter region of insulin gene and then transcribes the insulin gene into mRNA. The mRNA is then translated by ribosomes into protein, that is insulin. So here, if there is a mutation in the functional gene itself, let's say the mutation is here. So what happens if the gene itself is mutated? Either the protein that is produced will be defective, or there will be no protein produced at all. This is one scenario. Now, if the mutation is present in the promoter sequence itself, what happens is usually the RNA polymerase is unable to bind to the promoter region. Hence, the insulin gene is not transcribed, nor is the insulin protein made. This mutation will lead to no insulin production. Another scenario is there is a mutation in the ribosomes that are making the protein. Even in this case, the protein that are produced are either defective proteins or there are no proteins produced at all. So at the cellular level, depending on where the mutation is present, it will all affect the product that is formed from the gene at the end. It could either be a defective product or there could be no product at all, which will all lead to some kind of diseases. If mutations could lead to significant damage that could cause cancer, why is there any need of mutation? Why would natural selection retain mutation? The answer is that mutation is a reason for genetic diversity among organisms. Mutations, favorable or not, introduces variation in a population. This creates a variety of phenotypes that help organisms to adapt to an environment that drive evolution. The drawbacks of mutation is that it could lead to unfavorable phenotypes such as cancer, albinism, or any metabolic diseases that affect the survival of organisms. Thus, there should be a fine balance between mutation and its repair. The sources of mutation are replication error, damage to genetic material, and transposons. During replication, DNA polymerase reads the template strand and makes the new strand by adding new nucleotides that are complementary to the template strand. Whether the added nucleotides are accurate is read by the 3' to 5' exonuclease activity of the DNA polymerase. Even then, if the nucleotide that is added is wrong, it leads to mutation, which is then carried by the daughter cells if the repair mechanism of the cell does not recognize this mutation and repair does not take place. Damage to genetic material. Damage caused to genetic material will also result in mutation. Physical damage. High energy radiation causes damage to genetic material, thus causing mutation. Certain chemicals can also react with DNA, causing mutation. This causes chemical damage to the DNA, which leads to mutation. Transposons are also called jumping genes, are genes present in the genome that can change its location from one region of the genome to the other. This also could lead to mutation if the transposon inserts itself inside a region of a functional gene or its promoter. Let's consider this as chromosome 1 and chromosome 2. And our insulin gene is present on chromosome 1. And a transposon is present on chromosome 2. What happens spontaneously is transposon removes itself from chromosome 2 and inserts itself in between insulin gene on chromosome 1. Now what happens is the insulin that needs to be produced is hampered because the transposon is in between the functional insulin gene. So because of this, there will be no insulin produced in the cell. So this is one type of mutation caused by transposons. Based on the causes of mutation, mutations can be classified as spontaneous mutations and induced mutations. Spontaneous mutations are caused by natural causes. That is, due to error during replication, spontaneous deamination or depurination caused by hydrolysis, or by the action of transposon. These are naturally present in the cell and thus are considered spontaneous. Whereas induced mutations are caused by exposure to mutagens such as radiation or carcinogens. So spontaneous mutation is important in forward genetics and induced mutation is important in reverse genetics. Forward genetics is the identification and characterization of the gene that is responsible for the mutant phenotype. That is, once if you have the nucleotide sequence of a gene, and if you look into other genes that could be defective, you can predict that this gene is responsible for causing a disease in this phenotype. Whereas in reverse genetics, it is an experimental molecular genetics technique that enables researchers to elucidate gene function by examining changes in phenotypes or organisms. So if you know a particular gene is responsible for a particular function, you either mutate the gene on purpose or you cut it out of the genome and check the phenotype of the mutant which is resultant of such a mutation. Then the changes that are produced due to this mutation will tell you the importance of the gene in that particular function. Let's say you mutate a gene sequence that is responsible for hair color. Once it is mutated, you check in the progeny what is the color of the hair. If the hair color has changed significantly, it means that the gene that you mutated is responsible for hair color in the progeny. Discussing further on replication errors. If this is the parent strand, each of the strand is used to make the daughter strands. So here, instead of T that has to be paired with A, the G has been inserted. Usually at this step, the cell repair mechanisms will come and replace this with T. If it does not continue, then after second round of replication, the G is continued in the daughter cells, which will result in mutation if not repaired by the repair systems that are in the cell. So all the cells that carry this mutation will further divide into other cells which carry the same mutations, whereas these are the cells that have the normal copy from the parental type. So these are the consequences of errors during replication. So if there needs to be a correction of mutation, it has to take place before second round of replication to save the cell from mutation that could happen if it is not corrected. Base substitution is the simplest form of mutation. This leads to change of base present in the sequence. There are 4 types of bases that are commonly present in the DNA. This is adenine, guanine, thymine, and cytosine. Adenine and guanine are called as purines, whereas thymine and cytosines are called as pyrimidines. If a purine is exchanged with a purine, or a pyrimidine is exchanged with a pyrimidine. This kind of mutation is called as transitions. If A is replaced by G, or if G is replaced by A, this is called as purine to purine transition. If T is replaced by C, or if C is replaced by T, this is called as pyrimidine to pyrimidine transitions. So there are only 4 possible mutations that can take place in transition. In transversion, pyrimidine is converted into purine, or a purine is converted into a pyrimidine. So if A is converted to C, or if A is converted to T, it is called purine to pyrimidine transversion. If G is converted to T, or if G is replaced by C, it is also called as purine to pyrimidine transversion. Or if T or C is replaced by either A or G, it is called as pyrimidine to purine transversions. So there are 8 possible mutations that can take place using transversion.